Chapter 4, flip notes. Make sure you read pages 51 through 54 and you have these slides completed before next week's class. So you should have them printed out. You should have the first part of these slides filled in and ready to go on Saturday. So we're going to learn about cells in this chapter, and they are the simplest collection of matter that can live. We know that the cell is the smallest functioning unit of life. Cells were first observed by Robert Hooke in 1665. Working with more refined lenses, Anton van Leeuwenhoek was able then to actually look at and observe blood, sperm, and living organisms that he had found in a local pond. So we have a variety of different types of microscopes that we can use to see organisms that are too small for our eye to observe. And we used one last week called a compound light microscope. So what a light microscope does is it actually uses light as it passes through the specimen. So it goes through the specimen and then through a lens that magnifies it. So you can kind of think of it as just being a very fancy magnifying glass. Now, for most of our light microscopes, we can magnify specimens up to 1,000 times the actual size. Generally, that is with the aid of what we call oil. We have certain lenses that we can use oil to help with some of the, I don't want to say aberrations, but some of the irregular light bending that we get when it just goes through air. So again, it's not something that we'll probably observe in class. I don't think we'll use the oil immersion lenses, but we can get up to 1,000 times with the aid of oils. Now you should definitely know the difference between magnification and resolution. So of course magnification is pretty easy. It increases the apparent size of an object. Now resolution is the clarity of the image. You'll notice that as you magnify, you start to lose your resolution, and that is the limiting factor with the light microscope. Many of you guys have probably experienced this when you've cut and paste pictures off of internet and you put it in the document and you try to blow it up bigger, and as you start to blow it up bigger, it starts to get fuzzy. And so that's that resolution issue that we have problems with. So that's the difference between magnification and resolution. So like I said, the light microscopes have limitations. The human eye and the microscope, they both have problems with resolution. So, and that again is the ability to distinguish between small structures. So again, we have limitations with our light microscope, and therefore we're going to have to use another tool to see something smaller. And that is what we have been working on with technology. So with using light microscopes, we were actually able to study some of the microorganisms that we hadn't been able to see with the naked eye. We were able to see some very small animals and plant cells and some structures within the cells, which we'll refer to those as organelles. In the 1800s, these studies actually led us to the cell theory, which states that all living things are composed of cells and all cells come from other cells. Make sure that you know that cell theory. So obviously this slide did not transfer over well, but this is a picture that is in your book, and it talks about the size scale, which we did so last week in lab. So make sure you review your metrics and the prefixes and the sizes. And again, we usually will be dealing with our you need to know your base unit, which is your meters, and then we talked about millimeters, centimeters, micrometers or micrometers, and nanometers. So again, here are some of those units that I just mentioned in the last slide, and we went over this in the lab portion last week of class. So here's some of your conversion units here. So beginning in the 1950s, scientists started using a very powerful microscope called the electron microscope to view these very small ultrastructures of cells. And instead of using light, we actually use beams of electrons. And by using these electrons, we could see then how they behave and then be able to reconstruct a picture. So the resolve of these biological structures could be as small as 2 nanometers. Remember, that's 10 to the negative 9th. So that is a magnification of 100,000 times. There are two types of electron microscope. There is the first one, which is called the SEM, or scanning electron microscope, and what that does is it allows us to see the actual surface of that specimen that we're observing. And then the second part is called a TEM, or transmission electron microscope. And this one we're able to kind of like slice up our specimen and see more of the interior of those structures. So you can see here, this is a paramecium, and we can see the surface, we can see the cilia, we can see the oral groove, and so this is going to be our scanning electron microscope. Now here's our paramecium again, but in this picture we're using our TEM, our transmission electron microscope. And so we're actually able to see the interior structures and some of the organelles inside of our paramecium. The third type of microscopy that we're using today involves differential light and using what we call fluorescent dyes. So we're going to target certain molecules with chemicals that give off a wavelength of light that we cannot see with our naked eye, but the technology can. So this is a different type of microscope, and you can see the amount of detail we can get from this new type of differential interference micrograph. So we can see another little problem here with our line, but we're going to talk about cell size and why cells are small. They need to be big enough in order to house the DNA, the proteins, and the structures that are inside the cell. They need to be able to survive and reproduce, but they also have to stay small enough to allow for what we call a surface area to volume ratio. And with our surface area to volume ratio, that's really important when we're talking about having enough surface area to exchange materials with the outside, to get nutrients in, waste out, and then also to be able to move those materials within the cell in a timely manner. So we can see in this slide, if we had a big cell that's 3 by 3 and we figured out our surface area, if you remember, surface area is the length times width, and of course then in a cube we have 6 sides, so we'd have to times that times 6. And then over here we have broken up our cube into little tiny cubes, and those cubes are 1 by 1 by 1. And we can then figure out the volume, and the volume is going to be our length times width times height. And of course we're going to have to multiply it by the number of cells that take up that space. So we can see here our surface area to volume ratio for the big cell is only 2, but if we have a bunch of little cells occupying the same space, our surface area to volume is 6. And that's what we want. We want a higher surface area to volume ratio so it allows for efficient exchange of materials. So it's very important in why cells are very small, and we don't have these multicellular, or should I say, like a huge one-celled elephant. We have a multicellular elephant. It just doesn't work. And we're going to talk about diffusion and osmosis in a later chapter so you can see how this works. But right now you need to know that surface area volume ratio is kind of what governs or keeps our cell size small. So this ends the presentation for the first part of chapter 4. Make sure you have this completed for class, and then we'll fill in the rest of the blanks during class next week. Have a nice week.